Metabolic energy can be stored not only in the form of energy-rich bonds, but also by separating electric charges from each other using an insulating layer to prevent them from redistributing. In the field of technology, this type of system would be called a condenser. Using the same principle, energy is also stored or conserved at cell membranes. The membrane functions as an insulator. Electrically charged atoms and molecules, ions, function as charges. There are three ways of energy conservation at membranes: electrochemical gradient, proton motive force, and energy conservation in proton gradients. First up is electrochemical gradient. Although artificial lipid membranes are almost impermeable to ions, biological membranes contain ion channels that selectively allow individual ion types to pass through. Whether an ion can cross this type of membrane, and if so in which direction, depends on the electrochemical gradient that is on the concentrations of the ion on each side of the membrane, the concentration gradient, and on the difference in the electrical potential between the interior and exterior, the membrane potential. The membrane potential of resting cells, resting potential, is minus 0.05 to minus 0.09 volts, which means there is an excess negative charge on the inner side of the plasma membrane. The main contributors to the resting potential are the two cations sodium and potassium, as well as chloride and organic anions. The behavior of an ion type is described quantitatively by the Nernst equation. Delta G is the membrane potential in volts V, at which there is no net transport of the ion concerned across the membrane, which is equilibrium potential. The factor RT over Fn has a value of 0.026 V for monovalent ions at 25 degrees Celsius. Thus, for potassium, gives an equilibrium potential of minus 0.09 volts, a value the same as that of the resting potential. By contrast, for sodium ions, delta G is much higher than the resting potential, at positive 0.07 volts. Sodium ions therefore immediately flow into the cell when sodium channels open. The disequilibrium between sodium and potassium ions is constantly maintained by the enzyme sodium-potassium ATPase, which consumes ATP. Next up is proton motive force. Well, hydronium ions can also develop electrochemical gradients. Such a proton gradient plays a decisive part in cellular ATP synthesis. As usual, the energy content of the gradient depends on the concentration gradient's means on the pH difference between the two sides of the membrane. In addition, the membrane potential also contributes. Together, these two values give the proton motive force delta P, a measure for the work that the H-positive gradient can do. The proton gradient across the inner mitochondrial membrane thus delivers approximately 24 kilojoules per mol H-positive. Last one is energy conservation in proton gradients. Proton gradients can be built up in various ways. A very unusual type is represented by bacteriorhodopsin, a light-driven proton pump that various bacteria use to produce energy. As with rhodopsin in the eye, the light-sensitive component used here is covalently bound retinol. In photosynthesis, reduced plastoquinone, represented as QH2, transports protons, as well as electrons, through the membrane. The formation of the proton gradient by the respiratory chain is also coupled to redox processes. In complex, a Q cycle is responsible for proton translocation. In cytochrome C oxidase, complex 4, H-position transport is coupled to electron flow from cytochrome C to oxygen. In each of these cases, the H-positive gradient is utilized by an ATP synthase. 4. To form ATP. ATP synthases consist of two components, a proton channel, F0, and an inwardly directed protein complex, F1, which conserves the energy of backflowing protons through ATP synthesis. So that was all about energetic coupling. Keep watching sqadia.com for more.